You golf player?
Only when he has to be. But sometimes he has to take clients golfing. So he's got to do that, I think, next week or the week after. I think he's got to go to Oklahoma and take people golfing. He does not enjoy it, but it's kind of just one of those things he has to do for work. Usually he takes people out to dinner and drinks and things like that. And sometimes they do fun activities, taking clients like mountain biking and stuff like that. That's fun too. Like he'll do that because he's a cyclist, but he only golfs when he has to, which is funny because we live in a golf neighborhood. Does your neighborhood have golf in it? I thought it did. Yeah. Because you're just over across the street, right? Yeah, you guys have golf. Yeah, we've walked onto this golf course once or twice to look at it, but we've never golfed. I've never golfed. You're gonna golf here?
No, we're not allowed to go on.
Oh, you're not allowed to. Yeah, we're not allowed to either. We did it anyway, though. We just walked down the sidewalk and just looked. Okay, look, and then walked back. Yeah, you're not supposed to be on it.
Snowing.
Uh-huh. Yep.
Yeah, we went on a golf course.
Yeah, everybody in this neighborhood does that too. You're not supposed to go on the golf course, and you're certainly not supposed to go sledding on the golf course, but every time it snows, all the neighborhood kids will absolutely go sledding on the golf course. I'm sure we do a decent amount of damage to it, but I don't think my kids have ever been on the golf course sledding because we have a little bit, our neighbor has a little bit of it. I mean, we have a hill, but we have a lot of trees. Our neighbor's cut out some of his trees, so he had people sledding down his hill a couple of years ago when we had really good snow. And he seemed to enjoy it, so that was good enough. How about you, ladies? Did you have a good weekend? Anybody do anything fun? No? Have you started rehearsals yet for your show? Yes. Very good. Very good. Yeah.
Reading pages of a script right now,
Mm-hmm.
and they're just sleeping.
That's all, huh? That's not a lot. 3 pages.
Just for auditions.
Oh, I thought you already auditioned though.
For a different one.
Oh, for a different one. Oh, what's the other one? Yeah, Little Mermaid and something else, right?
Wasn't
Grinch. Okay, very good. Well, that sounds like fun. Sounds like fun. Well, hopefully, rehearsals will pick up soon and you'll be busy and having fun and all of that. A good deal. Very good, very good.
How my old private school, I had to I didn't really we had to do shows every year.
Oh yeah.
And then, I didn't really want to do anything.
No, they made you do it anyway, huh? Where did you go to school?
From school.
Okay, yeah. So everybody had to do like
Apartment.
the production.
We would write which at least 3 people of which one we wanted.
Mm-hmm.
They would choose it.
Yeah, they'll pick for you. Yeah, you don't get to pick. Yep. Oh no.
My last year there, I had to do narrative.
Oh, that's so every year at our church on Good Friday we do the Passion, but we it's chanted, it's sung. So it's some churches will do it in Latin, we do it in English, but I do the narrator part every year. So it's only 3 people. One person does all of the lines that are spoken by Jesus in St. John's Passion. The other person does any line that is spoken by any other person. So it's like Pontius Pilate and, multiple other people. Peter, that person does all the other people. And then the narrator does all of the narrative that is all of St. John's Passion. And I do that. So it's about 17 minutes of straight chanting for me. And everybody else finishes when Jesus dies, everybody else finishes, but I have to keep going because I have to finish the end of the Passion. So everybody else is silent and it's just me singing by myself for like an additional 3 or 4 minutes. It's really kind of wild, but I've done it every year now. So it's the same every year. Yeah, it's St. John. So it doesn't change, but it's St. John's Passion every year and I do the whole thing every year. It's long. It's like kind of one of those where you stand up and you're like, okay, here we go. And then you just start and you just keep going and keep going and keep going. I like it though. I really like doing it. It's one of my favorite parts of the year because it's very difficult. Like I have to say the hard things. I have to say all of the hard and terrible things that happen in the story because that's the narrative. So I have to tell that part, but it makes like it makes Holy Week real for me. Like that's I really like that because I'm forced to say that everybody else has to listen, but I'm the one that has to force myself to say it. So I have to say things about, like Jesus was whipped, he was hit, he was crucified. I got to say all the words. I have to say all the words. So every year it just makes Holy Week very real for me when I'm the one saying all of it every year. Well, good. I'm glad everybody had a good weekend then. I got these are new. I don't think you guys were doing it as much, but I got new cups and I put them up on the Skittles machine. Some people were like opening the drawer to the chest over there and taking out the bigger cups for their Skittles. I don't want to do that because those are for like when we have parties or occasionally if I just need like paper cups for lab. These little cheapy ones are fine for Skittles. We don't want to use the bigger, more expensive ones for Skittles. So anyway, if you get Skittles and you need a cup to put them in, they're over there now on top of the um gumball machine. Okay, are we ready? Lesson 5? Yeah, sort of, maybe. You're thinking about being ready? Well, before we start, I have some really interesting news. So I put this in the forum. I don't know if anybody saw it, but last year at the end of chemistry, we discussed a story, mostly with the lab rats, but I think we touched on it with you guys a little bit. And there was a huge breakthrough, big development in that news story that we discussed last year. So I wanted to share it with you. For those of you that did chemistry and you didn't, but you can still follow along, this is interesting. Do you remember anything about Bear Brook? We talked very briefly about Bear Brook. So there was this child called the middle child. So the reason we talked about Bear Brook, mostly with the lab rats, but a little bit with you guys, is because it was a situation where they brought in science to try and help solve a mystery, an actual crime. And one of the things that they used at that time was isotope testing. So what had happened was about 50 years ago, they found a barrel in some woods in New Hampshire with some bodies in it, 2 bodies, a woman and a baby. And then, or a very small child. And then a couple years later, many years later, actually, almost 10 years later, they found a second barrel with 2 other little girls in it. And they believed that they were all killed by the same person. But the problem is they didn't know who these little girls were or who this woman was. So one of the things that they were doing to try and identify them was they took their teeth and their bones, and your body absorbs whatever's in your environment. So there are isotopes, which if you remember, isotopes are atoms that have a different number of neutrons than your standard atom. And some isotopes are present in certain geographic regions more than in others. So they can take the teeth of somebody who's unidentified and they can say this person grew up in the northeast part of the United States. And then they can, because whatever's in your enamel when your teeth form when you're very small, like your enamel in your teeth form when you're very small, even your permanent teeth, while they're still, before they've erupted, your enamel's forming. And those isotopes get in the enamel. And then your hair, though, the isotopes in your hair show where you've been more recently. So they were able to tell like where these people grew up and then where they lived shortly before they died. So that was trying to help them narrow their search for who these people were. And then eventually they did some DNA and they found out through genealogy who 3 of them were, but there was one little girl that they could not identify. And they called her the middle child. Well, so last year towards the end of chemistry, we watched a video. I don't think you guys watched the video, but we talked about it, about Bear Brook case. The park was called Bear Brook Park. That's where they found them. Where 4 unidentified bodies in barrels were in a wooded area of New Hampshire. And we discussed how the scientists use isotopes in their teeth and in their hair to tell where they may have come from. The technique helped narrow down the identities of 3 of the victims, and then the 4th was eventually confirmed with DNA. But this middle child, they didn't know who she was. One thing they did find out about her based on DNA, once they figured out who those people were, who the 3 people were, they did eventually figure out who killed them. And one of the things they learned is she is the daughter of the man who killed all 4 of them. But they didn't know who she was or who her mother was for 50 years. So they knew that Terry Rasmussen, he's the killer, was her father, but that's all they knew about her. But now they know who she is. just this weekend. After 50 years, they found her identity. Her name is Rhea. So now we know that Rhea has a name, and DNA Dope Project did it. So I was going to read you this press release because it's really interesting. And it's written by this guy. See, it says Jason Moon. He's the guy who did the podcast Bear Brook for New Hampshire Public Radio, which is a really, really good podcast that talked about the entire thing, all the science involved. It was really interesting. So just because it's very science applicable and it's good science news, I wanted to share it. So New Hampshire Attorney General's office says it has identified the last unknown victim in the high-profile cold case commonly known as the Bear Brook murders. Rhea Rasmussen was just a few years old when she was killed by her father and then discarded in a barrel near Bear Brook State Park between 1978 and 1981. Previously referred to by investigators as the middle child, she was one of 4 victims in barrels found near the state park. Discovery led investigators, here's the crazy thing, to her mother, Pepper Reed, who was missing and had not been seen for decades, and they think that he probably killed her too. They had to find out who her mother was to find out who she was. And then once they found out who her mother was, they realized her mother had been missing for 50 years as well. So we still don't know where her mother is. So the case baffled investigators since the discovery of the first barrel 40 years ago and drew national attention as one of the first to be solved using a new technique called genetic genealogy, marking a turning point in how cold cases are investigated. Next quarter, when we do our big cold case mystery, you're going to be doing the genetic genealogy. So this is a case that just this weekend was solved using the same method that you're going to use for our mock cold case. So the technique was used to identify the suspected killer, whose name is Terry Peter Rasmussen, and 4 victims. The first, the adult woman, was Marlise Honeychurch. The other 2 girls were her daughters, Marie Vaughn and Sarah McWaters, and now Rhea. The case has weighed on New Hampshire and the nation for a decade, said Attorney General John Formella. And he said, with Rhea Rasmussen's identification, all 4 victims now have their names back. So it was DNA Doe Project that helped with this, which is a great organization. And that right there is her mother. And here's the crazy thing. They had to build a family tree using DNA from the little girl to try and find out who she was related to. They had a family tree with 25,000 people in it. That's 18 months worth of research it took them to get to her mother. And that's her mother. Her mother's name was Pepper Reed. And once they found her mother, they were able to find the baby, Rhea, the little girl, within 30 minutes. Because once they had her mother's name, they could pull up a birth record, and they pulled up the little girl. So that's how it works. So we're going to be doing something similar to that next semester. So anyway, that's the podcast. It's very good. If anybody ever likes to listen to podcasts, that's a really good one there. But it's just kind of crazy that we talked about this story last year. It used the method we're going to use this year in lab, and it got solved over the weekend. I was so excited. All right, so back to our lesson. So let's talk about characteristics of living things. All right. Oops. All living things are made of cells. What do you call organisms that are made up of only one cell? Do you know? There's a word for only one cell. Single-celled organisms are called unicellular. What's a unicycle? One wheel. So what do you think uni means? One. How about unity as one, right? Uni. So unicellular means one. And what do you call organisms that have many cells? If you don't know, what would you guess if unicellular is one? Multicellular would be many cells. Are you unicellular or multicellular? Yeah, we are very multicellular, aren't we? We have many, 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 many cells. So what do you call something that is a change in an organism's surroundings? Do you know? Do you know, Everly? She's looking. Stimulus, yes. So a stimulus is going to be your singular, stimuli will be your plural here. So if I turned the heat on in this room, that would be a stimulus. And you would get really hot. And then what might your body start to do? If you get really hot, what happens? What do you do? What's your body do when you get hot? You sweat, right? Okay. So that is a what? What would you call it? What do you do when you come in contact with a stimuli? What does an organism do? I mean, stimulus. What does an organism do? It's well, yeah, it's gonna respond is the word we want. So organisms respond to stimuli. So if I cranked up the heat in here, sweating would be your response, right? Or if suddenly there was a really loud noise, we all might go, huh, and that's a response, right? That's a response to a stimulus. All right, so then what is it called when an organism increases in size and becomes more complex? What do you call it? Are you getting bigger in size and becoming more complex? Develop's a good word. Yep. There's one other word for it, even shorter, easier, but grow is what we're looking for. So organisms grow by increasing in size and become more complex. And yes, development is growth. So we start out as one little cell, right? And then we become more and more cells. And then sooner or later, we become an old person. And those are all different stages of development, right? Organisms reproduce by producing what? What do you call it when an organism reproduces? What do you call the little critters it's produced? Yeah. You are your parents' what? Yeah. Your children. But you don't call puppies children, right? So what might be another word for that? Do we know? Offspring. There you go. Offspring is the word we want. Also a really good 90s alternative band. The offspring, right? Okay, so yes, organisms reproduce by producing offspring. So we have to consider the theory that life comes from life. Only living things can produce living things. Non-living things cannot produce living things. Even things that were once living cannot produce a living thing. It has to be alive to produce a living thing. But that's not always what we believed as people. We used to believe in the idea of spontaneous generation. So here's what would happen. Be back in the day before there was refrigeration, you'd get a piece of meat. You'd get like a big good cut of meat from your butcher and you'd carry it home and you'd put it on your kitchen counter and you'd walk away from it. And you'd come back a day later and it would be covered in maggots, or 2 days later and it's covered in maggots. And people used to think the meat produced the maggots because the meat was there and now there's maggots on it. But we know better now. Where do maggots come from? Do you know? Flies, right? So so here's the thing. What seems like common knowledge to us, maggots come from flies. Flies lay eggs, eggs hatch, maggots come out, maggots eat the meat, maggots turn into full-grown flies at some point, right? That seems really common sense to us. Like we're duh, of course, but that's not the way people always thought. People really thought that meat grew maggots. So you have to prove that wrong and show that it is something else in order to change the way everyone thinks. So for that, we can thank Francesco Redi. So we're talking 1668, which is really in the span of time, not that long ago. He was an Italian scientist and he had an experiment. So he took jars, all right, and he took these 2 jars and he covered one and then he left the other open, just like that at the top. And then he left them out for a few days. And then what he noticed after a few days is the one that was left uncovered got maggots, and the one that was left covered did not get maggots because no flies could get to it because it was covered. Seems like common sense. Revolutionary 1668. People go, oh, you mean the meat is not producing the flies? Oh, you're not making the maggots? What do you know? So this is one way that things that we look at today as just common sense were not always common sense. So spontaneous generation was the idea that a non-living thing or a once living thing, now dead thing, piece of steak, right, can produce a living thing. And we know that spontaneous generation does not occur. And here is another way it was like officially proved. So Redi did it first, and then we're followed by Louis Pasteur. Now here's something about this guy. That name, Pasteur. You ever heard the word pasteurize? pasteurized milk, pasteurized juice. Pasteurized is when you take something and heat it up for a moment to sterilize it and then put it in a sealed container, like milk has been pasteurized. It's they do that to get all the bacteria out so it does not continue to doesn't begin to grow things, right? You make it so that it is shelf more you'll last longer, not shelf stable necessarily, but that it lasts longer. It's a way of food preservation, right? And we get to thank this guy, Louis Pasteur, for pasteurization. So, quick side story. Odwalla juice. You ever had Odwalla juice? It's in the store. It's usually in the produce section in the little refrigerator with like the kombucha and stuff. So Odwalla juice, about 30 or 40 years ago, had a real problem. So they had orchards of apples, and the thing about Odwalla is that they used to advertise it as organic, non-pasteurized, fresh, like the freshest you could get was the idea. That they would squeeze the fruit, get the juice, refrigerate it, send it right to the store, and you'd be drinking like the freshest of the fresh. All right, so the apple orchards, if you pick the apples from the trees and then you squeeze the apples, and then, or you can wash them even a little bit if you want, whatever, you can run them through water, and then squeeze the apples and put the juice in. It'll last a little while before it starts to, it'll actually ferment, but you don't, but it'll last a little while. It'll do okay, right? But what they were doing on the apple farms is they were looking at the ground and they were seeing some pretty good looking apples that had just fallen off the tree that were on the ground. So they were picking those up too, the ones off the ground. Okay. And then they were running those through the process and putting the apple juice in stores without pasteurizing it, without heating it to kill any bacteria. No heating. Most of the stuff you eat has been pasteurized if you get it from the store. So the problem is people were not the only organisms in the orchard going after the apples. Deer were there too, and other animals that were running along the ground and picking up the apples on the ground to eat. And what else do animals do on the ground?
Eat.
Ah, yes. So there were bits of animal feces on the ground near the apples that they were picking up. Even if you can't see it, the germs, the bugs, the bacteria are still there. One of common bacteria that is in fecal matter is E. coli. Okay. So E. coli will make you very sick. So they were picking up these apples with E. coli on them. They didn't know that. And then making the juice. And multiple people, a lot of them very small children, were hospitalized. They were so sick because they were vomiting and had diarrhea to the point where they couldn't keep anything down. They got dehydrated. It was really, really bad. An E. coli infection is not fun. So there were kids that were very, very sick, and one might have even died. I'll have to look it up. But they were very, very sick, and it's because they were not pasteurizing their juice. So the idea of fresh sounds great, but I'm going to go with pasteurized any day. I don't want that mess, right? So in 1859, so 200 years later, Louis Pasteur set out to disprove spontaneous generation once and for all, and this is the experiment he did. He took beef broth and he boiled it in what's called a swan neck flask. That looks like that. The little whoop, see it? So he boiled it, and in boiling it, you sterilize it, which means you have killed any bacteria that live in it. You guys know that putting things in boiling water or boiling a liquid is going to kill all the bacteria in it. That's why, like, if you go camping and you get water out of a creek or a lake, you can boil it and it'll kill the bacteria that's in it, right? So he boiled it, and then he put it in this swan neck flask, which has a long bendy neck that traps dust particles and other contaminants before they reach the main part of the flask. So if the broth was boiled and the neck of the flask got broken after boiling and it was re-exposed to the air, it eventually became cloudy and gross, and they started to realize that bacteria was growing in it. So these are the ones that they didn't break, and these are the ones that they did break. And you can see how it looked different, right? So the experiment proved that there was no spontaneous generation. Beef broth does not produce bacteria. Beef broth will grow bacteria if it's allowed to get into the beef broth. So this is a video showing how it worked. Whoop. If it'll load. Here you go. Oops.
Video. Pasteur's experiment was designed to refute the spontaneous generation which was believed for a long time. More specifically, he used a special flask called the swan neck flask to challenge this fallacious theory. Firstly, he prepared 3 normal flasks. Beforehand, there's a soup in 3 flasks. Since soup contains a lot of nutrients, if you leave it for a few days, many bacteria will grow and give off an unpleasant odor. At the beginning of the experiment, the bacteria already present inside are sterilized by heat. This allows us to convince that the bacteria detected after a few days were formed after the experiment started. The tip of the first flask is opened and it will be left as it is. The tip of the second flask is heated and bent twice. This is what we call a swan neck flask. The biggest feature of this flask is that nothing other than air can enter the flask. By allowing only air to enter and preventing the entrance of bacteria, he tried to overcome the weakness of Spallanzani's experiment. Please refer to the previous video for Spallanzani's experiment. Just like the second flask, he made third one a swan neck flask. However, after he heated the flask, he tilted the soup to allow contact with the outside. The result was as follows. Bacteria had grown in the first and third flask, but not in the second flask. Since all flasks were allowed to contact with air, this result suggests that life would not occur spontaneously, even in the presence of oxygen. There might be an argument that the swanic flask is a special structure to prevent new life forming, but because of the third flask, he also denies that the swanic flask is simply disturbing life forms. In this way, as a result of this experiment, Pasteur succeeded in almost completely denying the theory of spontaneous generation that has continued since BC.
So thank goodness for good old Louis. Good old Louis. All right, so organisms have needs. We all have needs. Needs versus wants. Is a new Tesla a need or a want? What do you think?
Sometimes it,
Sometimes a need.
because
Maybe an automobile's a need, but the type might be a want, depending on your job or whatever. Yeah.
like because my grandma can't drive, so
Okay.
the Tesla is like
The self-drive. So that becomes more of a need for her. Okay, yeah, there you go. So needs versus wants. So needs. Living things need water. Why do we need water? Why do living things need water? What's water do for us? Do you know?
hydrate it.
It hydrates us, yeah. And what's the purpose of hydration, do you think? Why do we need to be hydrated? It's so that we can dissolve chemicals. We eat all these chemicals, we breathe in chemicals, we touch it, there's chemicals everywhere, right? Chemicals don't mean bad things, right? Remember, chemicals mean anything. Oxygen's a chemical, carbon dioxide's a chemical, right? All the food you eat is chemical. That doesn't mean that it's bad. Chemicals aren't bad, but we need water to dissolve the chemicals. We also need an energy source. And why do you suppose we need an energy source? Yeah, you gotta have energy. Yeah. So autotrophs are going to create the food from a non-living energy source, and then the heterotrophs are going to consume those autotrophs. So we need a source of energy, which for most of us is going to be the sun, right? And the sun is going to allow the autotrophs to produce energy from a non-living thing that then other living things can consume. And we need a living space. What do we need to what's important about a living space? What do you think, Everly? Space to do stuff, and you also need shelter, right? Can we live up in the Arctic tundra? Just live out there on our own? No, we have to create some sort of a space to live in that's safe, right? So we need a living space in order to be safe. And then we also need homeostasis. Does anybody remember what homeostasis is? So homeostasis means a balance. A balance in the amount of salt between outside your digestive tract and inside your digestive tract, or the amount of oxygen in the air versus the oxygen in your body. Homeostasis. It's a balance, and we need homeostasis in order for our cells to function properly. Our cells, and then therefore our organs and our organ systems, will not function properly if there is no homeostasis. So let's talk about classifying. First off, the word for classifying in science, and we'll do a lot more of this as we go, but the word for classifying like what type of species something is taxonomy. And the guy that did it was a student named Linnaeus, so sometimes they'll call it Linnean taxonomy. And we'll talk more about that as we move through, like next semester mainly. But if we talk about levels of classification, this is where we start. And this is the whole Linnean idea. So species are going to be the most specific, right? And here's one way you can tell. Typically, but not always, typically, organisms of one species can only reproduce with organisms of the same species. There are instances where you can have interspecies reproduction, like mules come from donkeys and horses. They're not a separate species. They're a mix of a donkey and a horse. But you have to have the same number of chromosomes for that to work, which we will talk more about next quarter. So you have species, and then your genus, and then your family, and then your order, and your class, and your phylum, and your kingdom, and your domain. For example, we are not in the same species as dogs, are we? No, but we are in the same kingdom, because it's kingdom Animalia. We're in the same kingdom as dogs. We're in the same phylum as dogs, Chordata, which means we have a central nerve cord that runs down, in our case, within our spine. And then, but it's not, you don't want to ever go vertebrate versus invertebrate, because there are some animals in phylum Chordata that have a central nerve cord, but they don't have a spine. And that particular order that throws it is called chondrichthyes, and those are sharks and rays. They have cartilage skeletons, so they're not really vertebrates, but they are chordates. Okay, and then, but we are in the same class as dogs. Mammalia. We're mammals, right? Dogs are mammals. So here's how you can remember those. You know how we have King Henry died unusually drinking chocolate milk to remember our metric units. Well, for taxonomy, basic taxonomy, biological taxonomy, we use Dear King Philip came over for good soup. So dear is for domain. We have 3 domain. And this has changed since your parents went to school, by the way. This is not the same that they will have learned it. So it's different. It's different from when I went to school. Now we have 3 domain. We have bacteria, archaea, and eukarya. And here's the thing, these will, domain and kingdom will overlap a little bit, and you'll see what I mean. But then the rest of them are pretty much a straight line. So then we have king, and that stands for kingdom. And our kingdoms are bacteria, archaea, protista, fungi, plantae, and animalia. So fungi, plantae, animalia, those 3, and protista, those 4 actually, are all going to be under eukarya up there. But then archaea and bacteria, as you see how those are repeated, because there's some overlap. And we'll talk about what eukarya means. Philip means phylum, and we have about 31 different phyla, which will be your plural for phylum. Came is class. And what does the word dynamic mean? Do you know what dynamic means? In music, what are dynamics? You guys know? Right, loud and soft, right? So it's about change. So dynamic means change. So when I say class is dynamic, I mean it's constantly changing. This isn't set in stone. We find a new class of organisms, and we find that maybe something wasn't classified correctly and it needs to be moved into a different class of organisms. But we estimate there are over 100 classes just in the phylum for Animalia. So just amongst animals, we've got about 31 different classes. And then when we get to order, we've got about, that one's also dynamic, and we have about 450 within Animalia. And then family, we've got about 6,000. Genus, hundreds of thousands within Animalia alone. And then soup, there you go. Actually, this should be over 100. I need to correct that one. That one's not right. Don't worry about it. But anyway, soup is for species, and we have over 1.5 million in Animalia alone. So here's what I mean by eukarya. Remember we saw the word eukarya? Okay, eukarya is for eukaryotes. And prokaryotes would be your archaea and your bacteria. They're going to fall into prokaryote. Can you tell what there are lots of differences between these 2 cells, but can you tell what the biggest, most important difference is? What do you think? Look at them more closely.
One's a circle,
One's a circle.
the other is like a.
Yeah, it's like a rod with a flagellum. Look at the inside. What's different? What's the big difference? You see it? Can anybody tell? So if you look right here, you see the nucleus? And you see how it has like even a center called the nucleolus, but you see how the nucleus has a little membrane around it? Do you see any nucleus over here?
No.
So prokaryotes, which is archaea and bacteria, have no membrane-bound nucleus. Eukaryotes do. So eukaryotes have a membrane-bound nucleus right there. Prokaryotes have no membrane-bound nucleus. What are you, prokaryote or eukaryote? What do you think? Do you think the nucleus in your cells is within a membrane? Or no? What do you think? Yeah, you're eukaryote. And you can remember that by saying, you're eukaryote, I'm a eukaryote, we're all eukaryotes, you're a eukaryote, right? So we are all eukaryotes. Eukaryotes are higher organisms. Prokaryotes are very, very basic, simple organisms. All right, so domains and kingdoms. What are the domain then of single-celled prokaryotic organisms whose cells lack a defined nucleus and have distinct molecular characteristics that separate them from other bacteria? That's where our archaea comes in. So we have archaea and bacteria here, and then eukaryotes over here kind of on their own. So bacteria or eubacteria or eubacterium, or bacterium, by the way, is the singular for bacteria. A bacterium, bacteria. So these describe and differentiate any group of prokaryotic true bacteria over here from the archaebacteria over here. Bacteria are genetically and morphologically distinct from organisms classified in the other two domains of life. So they're different. These two types are different, even though neither of these two types have membrane-bound nucleus. That's always hard to say, whereas these do. And then lastly, your eukaryotes, or your eukarya, which is the domain, is contains any cell or organism that possesses a clearly defined nucleus. Remember, it's nucleus, not nucleus, right? It's not nuclear either. You don't have a nucleus in an atom. You don't have a nuclear war. It's all nuclear, right? Nuclear. The eukaryotic cell has a nuclear membrane that surrounds the nucleus, in which well-defined chromosomes or bodies containing hereditary material are located. So it kind of looks like this. You've got bacteria. This is kind of how things evolved, they think. So you have bacteria and archaea down here, and then protista. These are single-celled, but they do have a membrane-bound nucleus. And then you've got plants, fungus, and animals over here. So eukarya is subdivided into 4 kingdoms: protista, plantae, fungi, and animalia. So your fungi are mushrooms, but they're also molds, slime molds, things like that. Animals you probably would recognize, plants you would recognize, and protists are these little single-celled, so these are single cell, single cell, single cell. The difference is these guys have a nucleus with a membrane. These guys do not, right? Cool. So here's a quick video on classification with our favorite amoeba sisters, who are always so much fun. I think there's a commercial though. Yep. We'll get rid of this commercial as soon as I can. Yeah, Sherwin Williams.
There are a lot of animals that I've always thought are cool. Salmon, hairless guinea pigs, iguanas, silky phantom chickens. But one day in my 7th grade science class, my teacher told us that we would get to see one of the most amazing animals ever in class, a hydra. And I have to confess, what I imagined in my head and what was actually reality was a little different, but I'm kind of used to that. Turns out hydra are pretty cool. Hydra are animals that are very small, a few millimeters in length, and they live in fresh water. They can viciously attack and eat their tiny swimming prey, and they can reproduce by budding an identical offspring off of themselves. But if you saw them, you might not think they're an animal. What makes us categorize them as an animal anyway, and not a plant or a protist or a fungus? In fact, how do we classify in the first place? Well, this lets us jump right into our topic: classification. Taxonomy includes the naming and classification of species, and much of the credit for starting a formal classification goes to Carl Linnaeus. We'll get back to that a little bit later in this video. But back in his time, the 18th century, there wasn't a separate category for other organisms like protists or bacteria. They didn't know about detailed cell structure differences. They didn't know about DNA, so they didn't know genetic relationships that now can determine how we categorize organisms. And that's the thing about classification. As we learn more about DNA and therefore relatedness, how we classify organisms can continue to change. So first, let's look at an awesome hierarchy system mnemonic. There's a lot of great ones that you can find that people have shared online. We'd also love to share ours. Dear King Paramecium cares only for green spirulina, but we sort of have a thing for protists, and we realize that might not be as memorable. So let's take a look at each group of this hierarchy, starting with one of the most inclusive groups, domains. Domains. It's so awesome that all of life will fit into them. So there are 3 domains, bacteria, archaea, and eukarya. The domain bacteria is full of bacteria. They're prokaryotes, and therefore they have characteristics we've mentioned of prokaryotes before. These can include bacteria that make you sick, the bacteria that are in your intestines helping you digest, the bacteria helping with decomposing, the bacteria fixing nitrogen in the soil, tons of different kinds of bacteria. Archaea are prokaryotes too, but they have some major DNA and structure differences that give them their own domain. And while they may seem more closely related to bacteria since they are prokaryotes, recent DNA evidence links them to having more in common with eukaryotes, which is interesting. Many of the archaea are extremophiles. That is, many of them like the extremes. Some like extreme salt environments, for example, which means they can handle extremely salty environments like the Dead Sea. Or methanogens. They can live where there is very little oxygen. In fact, most of them can't even handle oxygen. They use carbon dioxide to make their energy instead and produce waste gas, methane. Some of them live inside animals that eat a lot of cellulose, like cows or termites. Another extremophile is thermophiles. They like extreme temperatures. If you ever dreamed of living near the deep sea hydrothermal vents, well, if you were a thermophile, you'd be in luck. We mentioned that archaea and bacteria domains are separate because they have some major DNA and structure differences that are significant enough to separate them. And so does the third domain, eukarya. These are eukaryotes, and so they have characteristics that we've mentioned before that are common for eukaryotes. And that's where we'll focus right now. So the next level, less inclusive and more specific than domains, is the level of kingdom. Now here is the big disclaimer about kingdoms. Each organization is often changing, and it's not even something that all scientists agree on. We've seen a 5-kingdom system that looks like this, and a 6-kingdom system that looks like this. And it's important to understand it's a changing view as we learn more about DNA.
Your parents probably learned the 5. I did
Makes
too.
classification
Now it's
look
different.
exciting. But if we focus on these eukaryotes, let's touch on them briefly. Protista, extremely diverse, and there's often talk about dividing it because of how diverse it is. There are protists that are animal-like, and protists that are plant-like, and protists that are fungi-like, but many scientists don't consider them to quite meet the requirements to be in those kingdoms. Protista includes both autotroph protists, making their own food, and heterotroph protists, which can consume other things for energy. Most protists are unicellular, but they can be multicellular. Some have cell walls made of cellulose, which is kind of similar to plants, and some don't. Fungi are heterotrophs, so no photosynthesis for them. If that's hard to remember, just think about athlete's foot. It's a fungus on your foot, and it's not doing any photosynthesis there because that'd be really weird. Nope, it's there, causing irritation, eating dead skin cells. Fungi are usually multicellular, but they can be unicellular. And most of them have cell walls. Chitin, it's a carbohydrate. Next up, plantae. Plants are autotrophs. Yes, even the carnivorous plants, because they still make their glucose from sunlight energy. Plants are, for the most part, multicellular, and most have cell walls of cellulose. Finally, last up, animalia, animals. Okay, Hydra, you can come back. This mostly multicellular and heterotroph kingdom is the kingdom to which you belong. So now we have the other hierarchy levels. We get less inclusive, therefore more specific, as we move down to the Hydra's phylum, class, order, family, genus, and species. Because the species name is the most specific that you can get, it is the least inclusive. Now, you'll notice we wrote its species name here. Remember how we said we'd bring up Carl Linnaeus again? Well, it's because of him that we have this naming system, binomial nomenclature. This two-part naming system that we uses Latin or Greek roots. This is its scientific name. See that first name? That's its genus. It's written with a capital letter at the start, and it's written in italics. See the second name? That's its specific epithet, which is a fancy way of saying that it refers to one species in the genus. It has a lowercase letter, and it's also written in italics. So why do we care about these scientific names? Well, you can come up with a lot of common names for an organism that vary based on location. Take this mountain lion, for example. It's also known as a puma, cougar, or Texas panther. Same animal, different names. But its scientific name here is specific and recognized regardless of your location. And that gives power to an awesome way to organize and name species. Well, that's it for the new visitors, and we remind you to stay curious.
So we use binomial nomenclature. That's the two names, genus and species, to name organisms. So let's practice. What did they stand for again? Deer stands for domain. King stands for kingdom. Philip stands for phylum. Came stand for class. Over stands for order. For was family. Good was genus. Very good. And then soup was species. There we go. We got it. All right. So there you go. Good job. You wrapped it up and we are just about time. We got about 4 minutes. So yeah, get your lab ready. We'll bring everybody in a minute. I want to fix this one that I didn't like. Something was wrong with that. Okay, and then let me close this out.